Sylvain is a principal investigator in the Space Biosciences Research Branch at NASA and the GeneLab project manager. Sylvain has spent his career leading multidisciplinary research in both academic and government scientific institutions. He's the project manager coordinating the system's biology scientific research efforts for the GeneLab project, supporting both internal and external collaboration efforts, and the lead of the Radiation Biophysics Laboratory. So with that, Sylvain, I'll hand it over to you.
Well, thank you. Hopefully you guys can hear me.
Yes.
Okay, great. Thank you. Well, welcome everyone. I won't share my camera just because I've got some challenges with Zoom and my NASA computer, so apologies for this. Welcome everyone. I'll try to be brief so I don't get my 3 dings. I hope I can stay within 2 dings of this presentation. So let's dive into the Open Science initiative at the Biological and Physical Sciences Division. So next slide, please. You probably know that, but we are really, space biology is really focused on 5 elements listed here on this graph, you know, from microbes and plants to cells and molecules, animals, and developing organisms. And really, we're trying to do basic research to enable our knowledge of, you know, going towards applied science for human space travel. So this is kind of like the fundamentals of the work done by BPS, our division, and the open science effort. If you go to the next slide. And the open science, so for those of you who were at the ASGSR, you probably heard me speak a lot about this, but the open science resources at BPS really are, I would say, on the right of this, you can see the 3 databases that I think are critical. GeneLab is the database to collect and curate omics data. When we say omics, it's, you know, it can be genomics, it can be transcriptomics, proteomics, and so on. Anything that's not omics is actually archived in another database called ALSDA, for Ames Life Sciences Data Archive. And it has exactly the same goal, except we are looking now at curating mission data, project data, and, you know, imaging data are a big component of ALSDA. And then the last database, which is half a database, half a resource, is really NBIS, the NASA Biological Institutional Scientific Collection. And NBIS basically collects non-human specimen and space microbial cultures that are, you know, from experiments that were either conducted in space or experiments that were simulating some stressors found in space. In parallel to this, we have the BSP program, the Biospecimen Sharing Program. This is really a program that lets you, you can contact a BSP and ask for any tissue that were left over from experiment, and also work with them to basically, you know, basically this team dissect and preserve the rodent tissue from flight and ground investigation. And so there's a coordinated effort to share the tissue among as many people as possible. Next slide. So this is really a very brief overview of GeneLab. In terms of numbers, we have 387 data sets. We have 45 species. Like I said, we cover the full gamut of life, from human, microbe, rodent to plants and fruit flies. And as you can see on the top right, in terms of the organism, the majority of our data are space flight data. So, you know, 3D samples that were, you know, flown on the space station, on the space shuttle. It depends, you know, if it's archived tissue or fresh tissue. But having said that, there's a fairly large component of what we call ground studies, which are analog experiments. So this could be a bed rest study for human. It could be a Heimlimp unloading experiment for mice. It can do space radiation at NSRL, a variety of different experiments that are clearly relevant to what we do. The database is growing very fast. In terms of assay type, GeneLab is primarily focused on transcriptomics. It's not that we are focused on it, actually, it's because transcriptomics is really a very mature field compared to proteomics and other omics. And so the majority of investigators typically put a transcriptomic study in their grant these days. And so that's why we see so much of that. On the left, you see a snapshot of our, you know, the different aspects of the database. We'll dive into it. So if we can go to the next slide. So the database really, I don't think of GeneLab anymore as a database, but really as an enterprise. We provide so many services to to the scientific community. So you can come on our database in the repo, top left panel here, and discover the data we have. There you can mine and search very rapidly the information we have collected in those close to 400 studies. You can also contribute, and so that's the part on the right, top right panel. This really reflects the submission portal. So any PI can now submit their data through GeneLab through a very intuitive interface. We try to make it easy as possible for our PI to turn in their data, since it's now mandated to give your data to NASA once your grant is, well, as you build your grant work. On the lower left panel, you can explore through our visualization tool, and that's a little snapshot of the visual tool. And that basically lets you visualize process data from the raw data. This is a big deal. We spend a lot of our users are spending more time observing or mining the process data instead of mining the actual raw data. And then finally, on the bottom right, we have this component of teaching and education and outreach, where we basically provide analytical tools and tutorial to let you do your own analysis. And so this is a snapshot here of Galaxy, which is the platform we use for teaching students, and also you can have an account and run your own analysis. So in the next slide, please. So in terms of education, there's really a strong emphasis. You probably all are aware of the SLSTP, which is an undergrad student program that we have every year. And so there is an applications are open. This is the link at the bottom for those of you who are interested in having a professional experience in space life science discipline. We have the GeneLab for high school, which is at the top, and it's a 4-week intensive training program where rising high school juniors and seniors come and learn bioinformatics, really focused on space biology. So they get a chance to learn about space biology as bioinformatics. And there is the link for application there. If you click next, we should see the latest of our addition in terms of education. Is the GeneLab for colleges and university, GL4U. And that there is a course materials online if you want to check it out. And it's a boot camp, a 1 full week boot camp training, a bit more intense for undergrad typically. So, and this is something we're currently building in collaboration with many universities. So this is a rapidly growing entity of our education component. Next slide. So the analysis working groups, what are they? There's a couple of slides coming up there to introduce that. So we created these groups on January 25th, 2018. That was at the IWS, the HRP investigator meeting. And we went from, I think, 10 people to 150 people, scientists from multiple space agencies, international institutions, and industry. We meet monthly, and we have multiple groups that I will cover in the next. As a result, we publish over 10 collaborative peer-reviewed papers. Those are not GeneLab papers, they are AWG papers. I want to make sure people understand the distinction. This is the work done by the scientific community, with many of these scientists not even being NASA PI. It's just a very strong interest in what we do, to the point where we have all these incredible scientists, they're putting their own time in this and doing those fabulous analysis of what's in GeneLab and soon in ALSDA. So if you want to hear more about it, please join the breakout session today, and you can learn about it, and you can even sign up. So next slide. So there's 4 groups for GeneLab, one focused on animals. The number of members is listed in each of these panels. So I'm going to go fast. Animal, multiomics, plants, microbes. In a nutshell, the multiomics, by definition, is really looking at this holistic view of this response, a systemic response of these organisms, or that are exposed to some kind of space stressors. The animal group focuses more on the use of omics they focus on basic mechanism by which animals and tissue adapt to the environment. I think on the plants, astrobotany is a big aspect of this work. And for those of you who attended the SGSR, we heard multiple times through the TIDES effort from BPS that there's going to be a lot of emphasis on understanding how we can grow plants that can feed the astronauts when they start going very far from the Earth, and where supply is going to be very limited. And therefore, we need to find some ways of feeding these astronauts. So plants are going to be a big component, I believe, of the future work. Microbes, not to forget those, they're very essential. There's a strong emphasis on metagenomics, on metabolomics, and really, in my mind, the microbiome is involved also for the plants as well as for humans. So there's also this aspect of this ecosystem that involves the microbes and the host. So a lot of work there is done as well for these different groups. Next slide. For ALSDA, the AWG were just created in August of this year. ALSDA, in a nutshell, are data from Human Research Program and Space Bio. We're looking at phenotypic, physiological, behavioral level data, and also we are federating the data with GeneLab. There's about 100 members. Feel free to join us. Right now we've been focusing on setting up standards for the metadata. And we are also asking the team to provide feedback on the database and the tools. Next slide, please. So this is the charter for the AWG. Since I got the first ding, I better go fast on this. So we will share these slides if you're interested in the actual charter. I'm not going to read this, it's too long. And since I spent too much time on my previous slide, I will just encourage you to go check it out. This information is on the GeneLab website as well. Next slide. I think this is the next slide is also on the AWG. I think I should read a bit about this expectation for the AWG participation. Well, we want you to contribute significantly to the goals of the AWG, take part in group activities. Participants are expected to actively contribute to the group goals. And then there's a very strong emphasis on collaboration. The benefits of your participation is obviously multifold. A deeper understanding of space biology, but also the ability to write and co-author many landmark papers. So on the next slide. So this is the last slide, and that introduces the next speaker. So this is our agenda. Stefania Giacomello will be the next speaker. I'm not going to read all the speakers, but we are very fortunate. We're going to have a nice program. Enjoy the ride. Great work from the entire team. We're very lucky to have Sidson also giving us an overview of Leo Perrault. And then we'll hear more from other presentation on the different AWG emphasis that I discussed today. Not to forget the optional breakout rooms at 1:30 p.m. PST. So please make sure you stay. One last note, ALSDA is going to have also an AWG breakout room here. So that's kind of a new thing for us. So I hope some of you will be joining the ALSDA to discuss more how we can configure these new assays into the system. I hope my presentation was clear enough. And on this, I'll let, I guess, Stefania take over. Thank you, guys.
Perfect. Thank you. Thank you, Sylvain. Okay, I'm also setting an extra timer just in case. Perfect. Okay, hello everyone. My name is Stefania Giacomello and I'm a group leader at Cell Life Lab. Today I will present you a project that we are doing in collaboration with NASA GeneLab. And the project is about disentangling the impact of spaceflight on brain and heart by using spatial multiomics approaches. First of all, I would like to stress the fact, in case that we have some more junior persons in the audience, that the cell is the fundamental unit of living organisms. And this is valid both in animals and in plants. So that essentially means that cells in general, they organize themselves in order to create tissues. And then from tissues, we can obtain organs. This process then, it becomes in this process, it becomes very fundamental in which positions of the different tissues cells are in. Indeed, there is a paradigm in biology that considers how cells and their gene expression influences their locations in tissues and also how their location itself can influence their gene expression. Throughout time, we have seen the development of different type of methods in order to better understand cells in terms of where they are localized in the tissue and also their gene expression content. So relatively to the understanding of where cells are localized in tissue sections and to understand also better their morphology, histology has helped us very much in that sense. However, with the histological assessments of tissue sections, it's not really possible to understand the genetic program of those cells. So therefore, that type of information is completely lost. On the other hand, we have also bulk RNA sequencing, which has brought us a lot of knowledge based on gene expression that we can observe in samples. For example, if you have a condition or a wild type, if you are thinking, for example, of plants, and then you have a mutant, you can compare their gene expression and identify those genes that they are creating the differences in them. However, bulk RNA sequencing doesn't really allow you to get a very comprehensive view of all the differences that are happening between the different cells. And for that, in general, technology has brought us single-cell RNA sequencing, where we can understand the gene expression of each of the individual cells present in a sample. However, each of these three methods doesn't really allow us to combine both the histological information of cells, which is essentially their morphology and their localization in tissues, with their gene expressions. Either we have the morphological readout or the gene expression readout. So an ongoing question is, can we actually have a method that can combine both types of information in order to directly understand the molecular dynamics between cell location and a cell gene expression? The answer is yes, in the sense that lately we have seen development of many different types of technologies that are combining histological information with gene expression information. And one among them is the technology called spatial transcriptomics. It's important to underline that spatial transcriptomics is not just a technology per se, but is also the name of a whole field that can also be addressed as spatially resolved transcriptomics, which is really the study of gene expression in a 2D level in tissue types. So as I was saying, we also have this technology called spatial transcriptomics that we published in 2016, and we have also been able to extend it to plant tissues in order to understand those spatial gene expression patterns in different kingdoms. And very nice to note that the technology itself and also the other type of technology belonging to the spatially resolved transcriptomic field have been selected as method of the year by the Nature Methods journal in 2020. And that's essentially because of the power that this type of technology can bring in learning new biology and understanding the mechanisms in different tissue types of interest. How does the technology works? So I will go through some details about the technology, and then we will move on to towards the results of the projects that we are doing in collaboration with GeneLab. The spatial transcriptomics technology starts from considering simple glass slides, the same ones that you use, for example, for microscope analysis. The difference, though, is that the surface of these glass slides is divided into capture areas, which are these squares that you see here illustrated on this schematic. Now, if we look more into the details of how each of these individual capture areas is arranged, we can see that we can identify spots. These circles here that you see on the drawing, we call them spots. So these spots, they are then coated with probes, like in here in the middle of the slide, in which one side of the probe is attached to the surface, Then we have a spatial barcode, which is the key element actually of the method. This spatial barcode is a short stretch of nucleotides, which is spot specific. That means that all the probes in one of each of the different spots is the same. But if we start looking at spots of these capture areas, we will identify different spatial barcodes. And I will come back to this concept in order to make sure that we are on the same page. We also have a unique molecular identifier onto these probes, and the terminal part, the 3' part of the probe, contains a poly-T region. And this part is key in order to capture the polyadenylated transcripts in tissue sections. The surface of this capture area is around 6 × 6 millimeters, and we have about 1,000 spots in it. And each spot has a diameter of 100 microns. So now going back a little bit to the concept of the spatial barcode, if we consider a capture area like a XY space, like in here, in which each row and column of spots has its own coordinate, what we will see is that if we are considering this spot highlighted in, circled in red, we will see that the spatial barcode has a specific sequence, which also correspond to a specific coordinate, which is, for example, 4, 7. Then if we start looking at this other spot instead, we will see that the spatial barcode is different, as also the coordinate, and therefore for all the other spots on the surface. So it's important to remember that essentially each coordinate is associated to a unique sequence of the spatial barcode, but different spots have different barcodes. So the workflow then starts by considering any type of tissue of interest. It can be an animal sample or, for example, a plant sample. We section these tissues in order to obtain very thin tissue sections in order of micrometers that then we can attach onto the glass slide and specifically to the different capture areas. We then perform fixation in order to preserve the mRNA and the morphology of this tissue section and stain them in order to reveal the morphological structure that are in it. Afterwards, we perform imaging in order to really record first the morphology of the tissue of interest that we are studying, and also where exactly the tissue was placed onto the capture area. Afterward, we perform permeabilization in order to open up cells to transfer the RNA from the cells to the surface, in which we have the spatial barcodes and the probes that we have seen just before. And these probes on the surface, at that point, they can capture the RNA that is coming down from the cells through the poly-T region. And those probes at that point functions as primers to obtain the cDNA from the transcripts. And through this process, we are essentially barcoding each of the mRNA molecules in the cells with their own unique spatial barcode. Afterwards, we can prepare libraries from the cDNA molecules that we have collected from the surface for Illumina sequencing. After this data generation part, we can perform the analysis of our data, which consists in actually overlapping the imaging information that we collected at the start of the workflow with the gene expression information that we have been analyzing through sequencing. And by overlapping these two different types of information, we can obtain essentially a spatial map of the gene expression. And by looking at the image by using specific software, we can really go into the different part of the tissue of interest and seeing which type of genes are expressed in what part of the tissue domains. So a data structure of this type of technology, as I said, is two types, is both sequencing and imaging. Regarding to sequencing, we have an information contained on read1 that allow us to record the spatial barcode and UMI. And then the other type of information from the sequencing readout is transcript sequence per se, which is always coupled to the spatial barcode. While when we do the imaging analysis, we combine the image of the tissue that we have obtained with the positions that we have of the barcodes on the capture areas. And at that point, as you can see here, under the tissue, we exactly know where spots were localized. And since we know the coordinates of these spots with their corresponding spatial barcodes, we are then able to combine the sequencing information with it. And with this slide, I would like just to mention that there is a new version of the method, which has even more resolution, in the sense that in the same surface of capture area, which is 6M6 millimeters, instead of having 1,000 spots, we have almost 5,000. And the diameter of this spot is 55 micron instead of 100. So at that point, we really increase our spatial resolution that we can obtain for our studies. Now, coming to the type of project that we are doing in collaboration with GeneLab is, as I mentioned in the title, we want to study how spaceflight affects brain and heart. For this type of study, we had samples coming from 3 different mice that spent their time on ground, on Earth, and for them, and for us, these ones are ground control mice. And then we also have samples from mice that spent time on the International Space Station. So they were exposed to microgravity conditions and radiations as well. From these 6 mice in total, we have collected both hearts and brain. And for both type of organs, we are performing spatial transcriptomics assays with the new resolution, and also single nucleus multiomic assays. In that sense, we are not collecting the spatial information, but we are really able to look at the gene expression differences of all the different cells. And then by combining these 2 different type of readouts, we can perform spatial multiomics type of analysis. And why and also how do we do that? So the reason why we do that is because one cell in general, on average, has around 10 microns in diameter. However, as I just mentioned, the spot diameter is about 55 micrometers. So that means that we have more than 1 cell per spot. But we really want to obtain a spatial information of all the different cells present in our tissue. So then what we can do is to obtain spot cell type composition using the single cell data that I just mentioned. And that can be done through computational approaches. This is just one of them, is an example, in which we have the spatial data obtained with spatial transcriptomics. And here you see cells in this specific part of the tissue. And you see that you have multiple cells on the spots. And these cells can belong to different type of cell types. They don't really have to be belonging to exactly the same cell type. That means essentially that the gene expression profile of the spots will contain signatures for multiple cell types, all the ones that are in that spot. On the other hand, though, if we collect also single cell data from the same type of tissue, and from this type of single cell data sets, we can obtain gene expression patterns specific for each of these cell types, we can then computationally infer which type of cell types were present in the different spots. And in that sense, we can calculate cell type proportions of the cells that were covering each of the spots, therefore increasing our resolution. This is a way to, as I said, increase the resolution, but also, for example, to link RNA information with ataxic information, so the chromatin accessibility of those cells. Basically, we can use the RNA information that we can map back to the spot location and using it as a bridge to also obtain ataxic information at the spatial level. So now moving some of the results of this project. The first part is, or more than the first part, the first organ is the heart organ. And for in this case, what we did was to cut each of the hearts that we had into two. One half we have been sectioning it throughout the different levels, between 3 to 4, depending on the size of the heart. And at each level, we have collected tissue sections in order to perform spatial transcriptomics assays. On the other hand, on the other remaining half of the heart, we have collected the whole tissue, so we didn't process it at all, and we have extracted the nuclei from it in order to perform single-cell multiomics assays, the one that I just mentioned. For the brain, instead, we have analyzed with spatial transcriptomics only a specific level, which is the one that contains the hippocampus part in the brain. And at that level, we have collected sections for spatial transcriptomics and spatial mass spectrometry, while the other half of the brain has been kept intact in order to, again, isolate nuclei and use them as input for single-cell multiomics analysis. For the heart, we have collected 41 tissue sections that were basically half of them coming from the flight hearts, so from the mice that spent time on the International Space Station, and half of them from our ground control mice. For the brain, instead, we have collected 12 tissue sections in total, and also in this case, half of them come from the brains of the mice that spent time on the International Space Station, and 6 of them from the ground controls. What we see in this slide is a dimensionality reduction plot of the whole gene expression information contained in the spots throughout all the 41 tissue sections of the heart. And you see so many dots because, of course, we had cover almost 3,500 spots per capture area, and then we have to multiply that times 41. In total, we have almost 113,000 spots, and we also cover a very high number of genes, which is almost 23,000. Now, the color that you see here on the dimensionality reduction plot corresponds to different clusters that we can group the gene expression information from different spots in. And what we can do with the type of the technology that we are using is, the spatial location of each of those spots, because it was barcoded, we can map it back onto the original images that we have obtained. And here is an example for the heart, in which, for example, you start seeing one cluster present here in the ventricles and specifically in the trabecular portions of the ventricle. And also, for example, this darker cluster here present in the ventricles, but specific for a different tissue domain of the ventricles themselves. We also have spots specific for the atria, which are the 2 cavities in the upper part of the heart, as well as clusters specific, for example, for the adipose tissues or the vena cava and aorta part of the heart. In total, we have obtained 19 clusters, and this is a kind of annotation that we can do for each of the tissue sections that we have. Nevertheless, the power of the technology allows you really to analyze all the different sections at once. And here what we see is the spatial distributions of those clusters in all the sections that we collected for the flight and ground control samples. So again, you see, for example, in green that we have the atria, and in the bluish colors, the ventricles. The same type of analysis can be done for the brain samples that we have obtained. And in here, we have obtained 18 clusters in total which we can also map back again to the original samples, the original tissue sections that we have obtained. And as you can see it here, we have 6 sections here, up to here for the flight samples, and then other 6 for the ground controls. And also in this case, we can see that most of the clusters, or actually all of the cluster, correspond to the main anatomical domains that we can identify in the brain. For example, the cortex, the hippocampus, the thalamus, and the hypothalamus region. One of the first observation that we could do by looking at these global spatial gene expression patterns is that they don't really change between the 2 conditions, so between the ground control and the flight, neither in heart or in brain. And that somehow we can say that it was kind of expected, because if we think about it, also about the astronauts, like human astronauts, we do function on the International Space Station. So that means that the vital functions of the organs should be preserved. And this is essentially something that we could see here. However, what we can start doing is to start looking at specific differences between the small parts in the brain. And that means essentially for each of the different clusters. So for example, we could see that cluster numbered 0 was specific for the motor cortex part of the brain. And all the differentially expressed genes between the ground control and the flight samples for that specific part of the brain were specific for synaptic transmissions and mitochondrial processes. The same thing, we could do it, for example, for cluster number 5 that corresponded to the hippocampus region. And here we can see that the differentially expressed genes were specific for the neuroprojection development. Or we actually we can look at, for example, the striatum part here, where we have dopaminergic signaling in that specific tissue portion. And again, we could see that the genes indicated synaptic transmission alterations. So at that point, we have asked ourselves, or we have kind of complemented the information that was already originated by a study that was published last year with actually most of, or I guess almost everyone, yes, there was a part of the GeneLab Analysis Working Group of the Multiomics group, in which they have identified the mitochondria as a central biological hub for spaceflight impact. And in this study, we have studied different organs with different types of approaches, but not really being able to go down to the spatial level of the different organs. So at that point, what we can instead do is, for example, to start really zooming into the different mitochondrial alterations that we observe in the brain, and that I can also say that is valid for the heart, because we see mitochondrial alteration in the heart as well. But we can really see into which of the different clusters we see this alteration. And as you see it here in the image, if the mitochondrial processes are not present in all the clusters, but in each or in only certain specific portions of the brain. And how can we leverage actually this type of information? Well, for example, in the long run, we could try to develop specific drugs to cope with problems induced by space flight. And this specific drug can be for only specific of the brain, for example. So we can really go to a much finer scale of resolution. What then I can also say is that we have already fully generated the data set for our single nucleus multiomics analysis for both for the brain and the heart samples. And we have very recently obtained the full data set, which I can say that it was quite challenging to obtain. We have also already performed the setup of the computational pipeline for doing spot decomposition. So in order to understand what proportions of cell types are present in each of the different spots. And we have started to interpret the biology that we are identifying in the results. So please stay tuned. I can also mention that we have already started some validation of the spatial transcriptomics results that we have obtained, and that we are currently doing it with a spatial mass spectrometry for both neurotransmitters and lipid, because we are focusing also on the lipid metabolism, because it's another type of metabolism that we have been seeing changing specifically in certain part of the tissues that we are analyzing. Okay, perfect. And yes, so long-term plans. So this is one of the study that we are conducting, but the plan now is to extend this type of analysis to new organs, which include liver, femur, and the retina, or basically the eye, which are all organs that we know are quite affected by spaceflight. And overall, the even longer term plan is to understand the systemic response to spaceflight at the whole organism level across space missions. So not only in organs, but across different missions. And this is done like from my lab in collaboration with the Mason lab at the Weill Cornell, and of course GeneLab at NASA. I would also like to give some general comments about these studies that we are doing. So first of all, it has been quite challenging to establish all the techniques on the samples, because before the advent of these type of technologies, samples were collected and preserved in ways that were not really thought in order to, for example, preserve very well the morphology of samples in order to link the gene expression to their tissue domains. However, we start seeing with these results, the new omics approaches can really detect more subtle differences between ground control and flight samples. So there is a big need of standardized experimental setup, sample collection, and processing. And for that, we have initiated a new international consortium, which is called ISSOP, so International Standards for Space Omics Processing. And members of this consortium are working in collaboration with either NASA, ESA, the International Space, the European Space Agency, and JAXA, which is the Japanese Space Agency. And here is just an example of one of the focus groups that we are having. I'm basically done. Of, for example, how distorted was the morphology of the heart or the brain samples that we have obtained. So it was really important to, or we have spent a lot of time in order to orient properly these samples and make sure that we could capture at least essential morphological domains in there. And with that, I would like to say that we have already submitted a white paper for the NASA Decadal Survey in order to obtain or hopefully gain more power in terms of technological advances. And with that, I would like to thank everyone that has been working with us on the project. So Janetta, Osa, and Jakub from Scilab Lab in my group, as well as all the people from NASA GeneLab and the people at the Karolinska Institute that they have helped us to create nuclei from both heart and brain. And thank you for the attention.
Awesome. Thank you for that great talk, Stefania. I apologize for not introducing you earlier. I accidentally left the meeting. So let me just give you a quick shout out here. Stefania studied biotechnology at the University of Udine. At the end of 2018, she became a group leader at SciLife, and her research group focuses on the fundamental question of how cell localization influences biological processes in different kingdoms by developing and applying single cell and spatial transcriptomic approaches. So I apologize for that, Stefania, but we can go ahead and get started with our Q&A here. So we have about 3 or 4 minutes for Q&A. So let's start with this one. Is it possible to do spatial transcriptomics on slices from induced pluripotent stem cell derived organoids?
Yeah, potentially yes. So if because I mean in that sense now I don't know how well organoids they can preserve the full morphology of tissues, but as long as you have a solid tissue that you can work with, at that point you can perform this type of spatially resolved transcriptomics approach on them as well.
Awesome, thank you. Our next question is, do you have any plans to involve machine learning to classify the features obtained from image processing in the spatial transcriptomics?
Yeah, thanks for the question. Yeah, this is certainly one of the aims that we are targeting. So machine learning is going to be essential both for helping us interpreting the image data that we obtain, as well also to be applied in order to, for example, regress out all the technical variability that we can obtain that we can observe in our samples throughout space missions.
All right, cool. You're very popular. We got quite a few questions here. We'll see how many we can get through in about 2 minutes. Are there any differences in optimal section thickness among mouse tissue and plant tissues? So here's an example. Something too thick could cause lower resolution. Something too thin could end up losing RNA during staining. Also, plant cells are bigger with a cell wall, so it needs extra care in RNA extraction and bulk analysis. Do you have any special considerations?
Yes. Yeah, so what I can say is that this specific type of technology, at least, which is the one that I work with, can be applied also to plant samples, not just to animal samples. And in that case, yeah, I can say that it's also trickier to work with plant tissues. Nevertheless, it is doable. In terms of size, or like thickness of the tissue sections that we are using, is better to work with relatively thin sections, which can range between 10 to 14 microns, for example. So that always depends on the tissue type of interest. For this specific type of technique, though, in which we have a resolution of 55 microns, it's not so essential to have only one cell layer, because we will have a few cells in a spot anyway. So independently, if we have just one cell layer vertically, and yeah, but in general, I can say that the thinner the better, because that will allow you to permeabilize more efficiently the tissue section that you are working with. So if you start having, I don't know, a 30 micron tissue section, that will become very challenging to work with.
All right, thank you, Stefania. And we are out of time. I do apologize for all the other questions, but thank you so much, Stefania.
Thank you, my pleasure.
Awesome. Our next speaker is Lauren Sanders. She's a staff scientist at Blue Marble Space Institute of Sciences, and she's a bioinformatician working at NASA Ames GeneLab in the data processing and AI/ML groups. Her recent doctoral thesis at UC Santa Cruz focused on the use of comparative genomics and 3D organoid models to identify developmental origins and cancer drivers in childhood cancers. At NASA, her work involves using multiomics data sets and machine learning methods to characterize risks to human health from spaceflight travel. So take it away, Lauren.
Thank you very much, Lavorka. I think you can see my slides.
Yes.
All right. So I'm very excited to talk to you today about the future of machine learning in space biology. So to start with some background, just to make sure we are all on the same page here. So machine learning can be thought of as a type of computational modeling which learns over time to predict an outcome of interest from sample data by iteratively improving the model to minimize prediction errors. So we're going to talk about some applications of machine learning in biology and in space biology, but first, outside of biology, I think we are all familiar with myriad applications for machine learning. So for example, image recognition models train on huge data sets of input images and are then able to classify the image type. Text analysis, where a model assigns topic proportions and assignments based on text analysis. Recommender systems, which we probably all observe every day in our Netflix recommendations or our Amazon purchase recommendations. And all of these specific applications then are tools that, again, we're all familiar with. For example, the Google search engine leverages natural language processing, text analysis, image recognition. The Tesla autopilot uses artificial intelligence and machine learning to learn from huge databases of images and video, and road data and maps to make decisions. So what all of these examples, all these non-biological examples have in common is that they all take advantage of huge training data sets with hundreds of thousands of samples. In particular, all of these examples have more samples than features. So we'll discuss this more when we talk about the challenges of machine learning in space biology. But next, I want to highlight some of the advances of machine learning in biomedical settings overall. So one great example of recent innovation in machine learning and artificial intelligence in biology is the development of AlphaFold, which is a deep learning system trained on nearly 200,000 protein structures, which learn how to predict the physical structure of proteins based on amino acid sequence. And so the AlphaFold system is now able to determine protein structures with high levels of accuracy, and this is just a huge contribution to this decades-old famous protein folding problem, the problem of trying to determine the physical structure of protein based solely on its amino acid sequence. Now, a little bit more recently, during the COVID-19 pandemic, I think we've probably all heard, there have been many machine learning algorithms developed to diagnose COVID from lung scans or to identify the stage and the progression of the disease rapidly and with nearly the accuracy of a human physician or clinician. And so here I'm highlighting two studies, one which developed a radiomic signature of the whole lung to differentiate stable from progressive COVID-19 infection. And then this other study assembled and published a huge data set of CT scans so that the scientific community would be able to use this data set to train machine learning models. And so again, this study kind of highlights this need for large and high quality training data sets. The cancer field, of course, has been using machine learning methods and developing them both for image analysis and genomic analysis for several years. And so I chose three studies here to highlight from three different areas. So the first one is machine learning prediction of drug sensitivity of cancer cells based on genomic properties of the cell and chemical properties of the drug. The second paper here that I want to highlight is again using CT scan images to detect and diagnose lung cancer. And then finally, a machine learning technique to improve the prediction of survival in cancer patients using gene expression omics data. And so this can impact the different treatment regimen that a patient would be put on. So all of this background in different biomedical settings brings me to the challenge that I want to discuss today and that we're and trying to solve in different areas, which is we need to adapt machine learning principles to power knowledge discovery in space biology research. So as we all know, the space biology field shares many of the same aspects as these other biological fields, but we also face unique challenges. And so that's what I'm going to talk about next. So challenges for machine learning specifically in space biology. So let's talk first about some challenges with the data that we tend to work with in space biology. So the omics revolution, as we all know, has led to huge biological data sets which can characterize the most detailed molecular layers of the cell through assays like DNA and RNA sequencing and spatial transcriptomics, as we just heard in the excellent previous talk. So the thing is, though, in space biology, where experiments are necessarily small and expensive because they need to be flown to space or because they use expensive equipment and reagents, what ends up happening is this leads to high-dimensional sequencing data with tens of thousands of features, but only tens of samples. And so this can make it really difficult just to achieve statistical significance in comparisons, but then it also makes it difficult to apply out-of-the-box machine learning methods. A system with fewer observations than features is referred to as underdetermined, and models will tend not to converge satisfactorily. So additional work is needed to utilize these data sets, for example, reduction to select the most informative features. Then in space biology data, we also have the challenge of bringing together multiple types of heterogeneous data. For example, a space-flown mouse experiment may collect gene expression, mutation information, drug treatment, environmental variables like CO2 and temperature, and then histological tissue image data. So incorporating together all this heterogeneous and sparse data with missing values is both a challenge, but it's also an opportunity for innovation in machine learning and knowledge discovery. And then finally, this is not a unique challenge to space biology, but the need to transfer knowledge from model organisms to humans. Fortunately, we can learn from similar fields. For example, the biomedical field focusing on rare diseases faces many of these same challenges. The patient populations are small, they're spread out across different institutions and hospitals, and the data are sparse and heterogeneous. And depending on the type of disease, certain data types may or may not be available. So we can learn from machine learning innovation in this field. And I'm just going to highlight a couple of studies here. For example, X-Rare is a new method for prioritizing causal gene variants in diabetes. Another group developed a rare disease classification algorithm based on a knowledge graph of medical information, which is robust to the fact that the graph is incomplete. A group specializing in childhood cancer, which is a specific group of rare diseases, developed a method called multiplier, which uses a machine learning method called transfer learning to learn biological relationships from large non-rare data sets, and then apply that knowledge to the smaller childhood cancer data sets. And we could potentially imagine using a similar methodology to learn intrinsic biological relationships from model organism data and then apply to human data sets. And finally, a very recent publication on an artificial intelligence-based clinical decision support tool for identifying potentially causal genomic variants in newly diagnosed genetic diseases in infants. So with that background in mind, we can use our imagination and think about lots of applications for machine learning and space biology, both for repurposing existing methods to help us analyze our data and for us to develop novel methods built on these principles to solve the unique challenges of our field. So the next few minutes, I'm going to highlight specifically some of the recent developments in the work that we're doing in research, analysis, knowledge discovery, and hypothesis generation, AI/ML applications. But I just want to highlight quickly, I don't have time to go into this today, but the future of machine learning and space biology will also involve using machine learning for data management and for automation of experimental payloads and onboard analysis. So that brings me to a key example of an immediate application of machine learning to space biology data. So I want to talk about some of the work we're doing with a recently developed platform for causal inference in complex biological data, and this platform is called CRISP. This is an ensemble machine learning platform for causal inference developed by the Frontier Development Lab Astronaut Health team in 2020. So for those of you not familiar, Frontier Development Lab, or FDL, is an 8-week accelerated machine learning development challenge for space applications. So the CRISP platform, which came out of FDL 2020, one of the key things is that it is generalizable to different data types. And the idea is to be able to learn causal drivers of any feature within a data set, so you can input heterogeneous data. CRISP then uses automated dimensionality reduction to select the most important input variables. And then finally, this ensemble of machine learning methods votes on the selection of causal features. So the output of CRISP is features that are causal of a target variable. For example, genes that have a causal relationship with some disease state. One of the unique and powerful things about the CRISP platform is that it incorporates this idea of separate environments within a data set. So any identified causal features have to be consistent across all environments. So that makes it possible to synthesize data sets from different experiments, different labs, different missions, and ensure the robustness of causal results across separate environments. We employed CRISP to identify causal features within a space-flown mouse liver gene expression data set from a NASA GeneLab. We combined and normalized 4 different RNA sequencing data sets. And these samples also have oil staining data, which is really key here because we were able to combine 2 data types and take advantage of the heterogeneous data processing in CRISP. So oro staining, oil red O, this is the chemical stain that indicates the presence of fat-soluble lipids in tissue sections. And really key is that high oro values indicate the presence of non-alcoholic fatty liver disease. And based on previous research in these data sets, we believe that spaceflight may introduce an NAFLD phenotype, and we wanted to see which genes appeared to play a causal role in the appearance of this disease. So when we looked at the top 20 causal genes identified by CRISP, we do see that several of the top ranked causal genes are already linked to NAFLD in the literature, kind of validating the biological relevance of these causal genes. And when we performed gene set enrichment analysis to identify functional gene sets or pathways that are statistically enriched for those 20 causal genes, we found several pathways with relevance to liver, fatty acid metabolism, or the NAFLD disease phenotype. So these are still preliminary data, but these results do demonstrate that CRISP has indeed identified genes with the potential to be causal of an NAFLD disease state. We, during the FDL Astronaut Health Challenge this past summer, the team again used CRISP, but this time to evaluate the comparability of mouse model gene expression data with human data. So I was involved in this project as a mentor, and we combined 6 data sets from NASA GeneLab and from GEO, microarray gene expression, and 2 organism types, human and mouse blood samples, all that had been irradiated. We performed this set of CRISP experiments where we varied the amount of mouse data that we mixed into the human data. And then we compared those results to a run using only human data. And so in this plot here on the left, what you can see, each bar represents a single CRISP training run, where each run used a different ratio of mouse and human mixed data. And the y-axis is showing how many of the causal features overlap with the human-only run. So excitingly, these preliminary results do show that the overlap is never less than 50%, which was pretty robust. And then we also took a look at the 20 causal genes when we did a run with just all the human and all the mouse data mixed together. And we do see that these results, again, recapitulate the known relationship between radiation exposure and cancer. Several of these top-ranked genes are implicated in cancer progression. So although these data, again, are still preliminary, this first review of the data do demonstrate that CRISP can be robust to multi-organism data and enable this translation of findings between mouse and human expression data. The This team from this last summer also released CRISP version 2, which is updated with federated learning capabilities through Intel's open source OpenFL platform. Federated learning is a machine learning approach where a model can be trained separately at individual distant locations without actually having to share data. So this has implications for data that may be private and cannot be shared. And then only the updated model weights are released from each individual location and aggregated together into an aggregator. And then this training round continues multiple times. And so you end up with a global model that has been trained on multiple data sets, actually needing to share the data sets with each other. So again, this has implications for data that cannot be shared due to privacy concerns, or for data that are generated in space and cannot be downlinked to the Earth because they are too large. Finally, another key machine learning method that we found has relevance for space biology research is SPOKE, which is a biomedical knowledge graph that was developed in Sergio Baronzini's lab at UCSF. So a knowledge graph is just a graph-based representation of relationships between data, coupled with some type of reasoning algorithm that can traverse the graph to find new knowledge. And SPOKE incorporates over 30 human biomedical databases. And so the team that was published in a recent publication combined multiple RNA sequencing mouse clone data sets from GeneLab, integrated them into SPOKE to identify relationships with genes that were more highly expressed in spaceflight mice, and see which types of nodes they were most highly correlated with. And just to show a few of these results here, we saw some interesting symptom nodes, for example, space motion sickness, that are clearly relevant for spaceflight. So this brings me close to the end of my presentation, just to introduce the NASA Ames Artificial Intelligence and Machine Learning Initiative for Space Biology and Health. We're an interdisciplinary team with expertise in biological sciences, clinical application, and artificial intelligence and machine learning. And we focus on the development and deployment of AI/ML analysis methods to support research on the effects of radiation exposure and spaceflight stressors on life. And here I just want to highlight one of our key projects, which is the development of a model hub and analysis portal, which is going to be a user-friendly, publicly available portal to incorporate multiple data from GeneLab, starting with GeneLab FDA and then moving forward from there, visualizing the data set, incorporating models such as CRISP and SPOKE to perform automated analysis, and then an analysis portal with interactive dashboard of modeling and analysis results. So then finally, ultimately, we envision that this public modeling portal and the rest of the work at our AI/ML initiative will support the Biological and Physical Sciences Open Science Network, which collects data from both analog and space experiments, tissues, and ultimately makes all of these data available to the users through open science. So this is a collaborative culture that empowers the open sharing of data, information, and knowledge. And so ultimately, the key here is to have a worldwide network of users who are able to access the data and use the portals for training and for knowledge discovery. So this brings me to the end of my talk, and please reach out to me if you are interested in collaborating with our AI/ML space biology team, or if you know of or have developed a method that you think should be included in our modeling portal. We would love to hear from you, and I've put my email on the last slide. And finally, I do just want to acknowledge the work that's been done by everyone at NASA Ames, the UCSF SPOKE team, and the 2 Frontier Development Lab Astronaut Health teams that contributed to CRISP. And I'm happy to take any questions.
Wonderful. Thank you so much, Lauren. So we do have some questions for you here. Let's start off with this first one. This is a very powerful approach, and thank you for sharing. One question is, what types of missing data do you think could enable AI/ML methods better for space biology?
I mean, what types of data do we don't have now? like what's my wish list for data? That's how I'm going to interpret this question. I'm always interested in samples so that our models can be more powerful. And I think that one of the ways to do that could be incorporating experiments here on Earth or clinical data that we haven't yet realized are relevant and important for the comparisons that we're doing. So looking for creative ways to use the data we have.
Awesome. So here we have another question is, can CRISP handle heterogeneity in animal strains for a specific assay type? Example, RNA-seq data from C57BL6 versus BALB/C mice in the GeneLab datasets. If yes, how does it perform with separating biological variability due to strain differences versus exposure conditions?
Yes, absolutely. This is a great question, and CRISP can handle this, and that's because of the intrinsic environmental separation that is built into CRISP when you configure the experiment. So one of the things that you could do if you were interested in disentangling the signature from different mouse types or different exposure conditions, put into the CRISP config file that you have which samples come from different mouse types, but then use the exposure as either your biological variable or your target variable, and then that any causal features that come out of CRISP have to be robust and significant across those environments. So you know that anything that comes out is not related to one mouse strain or the other. It's actually consistent across both mouse strains.
Awesome. Thank you, Lauren. We have several more questions here. I think we have about 1 to 2 minutes left, so we'll see how many questions we can get through. But the next question is GeneLab planning to validate any of these AI/ML results using other methods? And what is the process for turning the GeneLab machine learning results into further experiments or countermeasures?
These are great future-looking questions. It would be great to be able to do some biological validation or laboratory validation of the findings that we're getting from these machine learning algorithms. That's something that we would like to do, but we don't have a structure in place for it just yet. And the other question was developing countermeasures. Obviously, ultimately, the goal of our work is to be able to be applicable to human astronaut health. But as the first part of this question suggests, extensive validation and experimentation would need to take place for us to be confident in suggesting some countermeasures.
Great. Maybe one more question here, and then we'll have to move on to the next speaker. Is it necessary to use quote-unquote space data for learning to build a model or gene expression simulator?
I am trying to understand the question. So can we use simulated data rather than directly having space-flown data? So I think that there is absolutely a place for synthetic or simulated data that can maybe learn from smaller space-flown data sets, but generate a larger, maybe more diverse data set without actually having to be space-flown. And I think modeling using synthetic data can absolutely derive interesting knowledge. One of the other projects that I didn't have time to talk about today is developing a suite of benchmarking training data sets where we actually are generating synthetic transcriptomic data in order to evaluate and benchmark future machine learning models. So I definitely think there's a place for that.
Awesome. Thank you so much. I apologize. We cannot get to the other questions because we are at time. And so thank you so much, Lauren. Great presentation. And our next speaker is Sidney Sun. He is the Deputy Director for Programs and Project Directorate at NASA Ames Research Center. Sid has been at Ames Research Center since 1988, endeavoring to create new aerospace systems while helping people and organizations grow to their fullest potential. Currently, he's the Deputy Director for Programs and Projects, and he provides management and oversight of Ames's cadre of project and program managers that are developing spaceflight missions and executing research and technology development projects. Thank you so much for joining, and take it away, Sid. Sid, if you're speaking, you are on mute.
Sorry about that. And can you see my slide right now?
We can see your desktop, but you're not in presentation mode.
Okay. All right. How's that?
We see you.
Looks like you need to start sharing again.
Okay.
Stopped.
What do you see now?
There we go. Looks good.
Okay, sorry about the technical difficulties. Anyway, thank you for giving me this opportunity to talk with everybody today. It's great to connect with the GeneLab community. I don't get to work with the biosciences very much now. I'm working more with projects like Sophia and Viper, but it's nice to be part of this once again. Big shout out to Sullivan and the GeneLab team. It's incredible what you all have been able to do with you really the goal that Marshall Porterfield initially conceived of, and how do we get the diversity and richness of data that we have with space biology out there to a broader community, and figure out how to mine this data, get more value out of it. You've all been doing that, so thank you. And so what I'm going to talk about today is an assessment that we did a couple years ago. I led a team of people, including researchers, engineers, scientists at NASA Ames, Macarena Parra, Steven Spremo, Fatih Karuria, Matt Lara, Hami Ray, and Tony Rico. We were asked by Space Biology, both Nikki Rail, who was the program manager, and Kevin Sato, who is the program scientist. They were recognizing that space biology was entering a new era in research. Most of the work that space biology had been doing at that time had been focused either on ground studies or on experiments on space station. With Artemis becoming more real, the gate getting more definition, and lunar lander projects being more defined, they saw some opportunities to do research beyond low Earth orbit and be able to study biology in the radiation environment more similar to what we'd seen on travels to Mars, as well as low and partial gravity environments. So they wanted to know what could space biology take advantage of in existing payload systems that could be perhaps adapted for beyond LEO experiments. Again, they wanted to focus on what is available through what could be used on Gateway, which is a small space station that NASA is building that will be orbiting around the moon, or the Commercial Lunar Payload Services, which is a series of landers that are going to be landing without people, but autonomous landers, and they'll be bringing payloads to the lunar surface. So we looked at what capabilities existed, where there were gaps in capabilities, and addressed strategies on how to close those gaps. So again, this study was done a couple years ago. I will note here that Matt Lara, who is one of our team members, is going to be updating this study. So it'll be interesting to see what more information, what new capabilities that will be added to this. But I'm just going to capture what we summarized a couple years ago. So 2019, as I was mentioning, the space biology research portfolio was either ground research or ISS, and then there was a smaller number of experiments that were taking advantage of small satellites, CubeSats, and then we have been flying periodically with the Russians on using the Bion and Photon space capsules. And so all of those space platforms provided a microgravity environment. In terms of radiation, because those platforms were basically flying in space within the Van Allen belt, they were seeing a radiation environment higher than what we see here on Earth, but not similar to what we would see on a journey to Mars. So what we were going to be able to get with experiments beyond LEO using Gateway or using CLIPS is different gravity environments and a radiation environment that's more similar to a Martian trip. And so with that we'd have a much greater understanding of how biology would be affected on trips to Mars, and perhaps project that onto our understanding or estimate of how human travel would be affected. So to support space biology experiments beyond low Earth orbit, we wanted to look at what existing systems do we have right now, and whether they could be adapted to work on these new platforms. So we looked at equipment that we were developing at Ames, or we have developed at Ames, as well as other NASA centers like Kennedy Space Center and Johnson Space Center. And then we also looked at hardware that's been developed by commercial vendors that are on use on Space Station. And then, of course, the international partners have developed a lot of equipment to study space biology. So we examined those systems. There were a few instances where the hardware has not flown yet, but they will fly soon. And so we thought they were worthy of including in our assessment. And then there were some hardware that had already flown, but were no longer in existence. But the designs for that hardware were still available. So we thought if for some reason those systems were needed, we could resurrect that hardware. So we did gather information through various ways. A lot of information is available publicly through the internet. And then our folks that were on the study have been involved in a variety of space biology experiments, some of them dating back to the shuttle era. So a wealth of experience on the project, and also knowing where there's different project documents that contain information. So we were able to harvest that as well. Where we had gaps in information, we did reach out to companies to fill in those gaps. We wound up filling in a massive spreadsheet. We had 78 different categories of information we were looking for, and I'll go over that in the next slide. So these are the different criteria that we were looking at, the 78 items. I won't go through that in detail, but I will summarize this in terms of some broad categories. So in the green vertical stripe, there are what I would describe the general descriptive information of the payload systems. So who developed the hardware? When did the hardware first fly? Do we know any costs associated with using the hardware? And then projecting on how these systems would be compatible with different platforms. How compatible would they be with Gateway? How compatible they would be with small sats or CLIPS landers. We evaluated those things in terms of technology readiness levels. And then in blue, recognizing space biology is a very broad discipline, many different aspects of space biology. We wanted to know what these systems were designed to address. Which aspects of space biology were they looking at molecular biology experiments or cell culturing experiments? Were they doing synthetic engineering, synthetic biology in space, or was it a lab on a chip experiment? All those criteria, all that bits of information were important to capture. And then in gray is showing what requirements associated with the hardware to operate the systems and to get scientific data back from those systems. So a number of the systems were designed to be crew-tended because they were built for space station where astronauts would be available. Some of them would be autonomous. Some of them were external payloads, required a robotic arm. It was good to know if that was needed. Some of the systems provided a capability to capture data on orbit, and we didn't need to bring samples back, but others were more reliant on bringing the samples home and having analysis of the specimens on the ground. In the peach stripe, we capture what I would call the engineering specs on the systems. So how large were these systems? What was the mass associated with them? How much power did they require? How much data did they generate, etc. And then returning to the science capabilities of the hardware, we captured a lot of information in what is shown in this yellow band. So we wanted to know more specifically what kinds of specimens were able to be sustained in these hardware systems, what environmental parameters were being afforded, were there different types of sensors in the system that could return valuable data. Some of the systems had a centrifuge capability, so that was valuable to know. Radiation measurements, temperature measurements, pH measurements, all of those are important to capture. We wound up looking at 64 different payload systems. I'm not going to show the complete list yet, just because it would be overwhelming, although the last slide I have does show those items. I'll just talk about it broadly here. We looked at systems that were able to grow different types of model organisms, either cells or microbes or plants. We did not look at rodents in this study because we heard from space biology that they did not anticipate studying rodents in beyond LEO in the next few years at the very least. We did assess different kinds of incubators as well as a variety of different on-orbit centrifuges that have been built. In terms of sample preservation, we did look at several different types of fixation, chemical fixation systems. We also looked at several different types of DNA, RNA analyzers, like the MinION that's pictured here. And then we also looked at on-orbit microscopy and plate readers, what capabilities were afforded. Other systems we looked at external payloads as well as internal payloads, so exposure facilities were an important cost to look at. Radiation clearly is an important measurement to capture, so we looked at different dosimeters. And then most of the experiments we looked at were for man-tended vehicles, but we did look at instruments and payloads that have flown on CubeSats. And it turned out the CubeSat payloads are very relevant to what we're talking about now for studying life beyond LEO. Understanding or making some assumptions on what lunar payload requirements would drive equipment selection, recognizing the CLIPS landers are going to be able to bring the equipment, the payloads to the surface, but there's no way to have any human interaction with those experiments, so the systems would have to be autonomous. Clearly they need to be small size, low power. Because of the radiation environment on the moon, they would need to have the electronics to protect from the radiation. And then there'd be a pretty broad range of temperatures that the equipment would operate in. It gets hot on the moon and it also gets very cold. So recognizing that small sats have been designed to operate in a very similar regime and in a similar way, we thought adapting the small sats to work with the CLIPS lunar landers made the most sense. In particular, there were at Ames, we had generated the BioSentinel mission PowerCell and ECAMSat. BioSentinel is an experiment that its prime mission is going to be launched on Artemis 1 in February, and it's a small sat that will be launched. It's a heliocentric orbit, and it's going to have different, it'll have yeast growing within the system, and we'll study how the yeast are affected by the radiation environment. That experiment is also being adapted to work on space station, and so another natural extension of this experiment would be doing a similar study on the lunar surface. Looking further ahead, we thought there's some other capabilities that have been developed for other applications, like the Europa Life Microscope, intended for an astrobiology mission, but its capabilities could be useful for space biology. It's at a relatively low TRL now, but it could, with some investment, it could be ready for flight and application in the near future. And besides instruments, we recognize that a variety of different hardware and electronics components could be developed by NASA to operate, be qualified to work on the moon. So things like developing radiation-hardened electronics, smart power, communications, data handling, storage. All of those would be a great value to the science community. Switching over to Gateway, which will be a small space station that will be orbiting the moon. Even though I described it as a small station and it'll have some of the similar capabilities, it'll operate differently than the ISS. The Gateway will be human-tended for 1 month out of a 12-month period. And during that 1 month when crew are on board, they're going to be much more focused on the human landing system, sending people back and forth to the moon. And so the time that will be available to support science operations will be very small. We're hearing on the order of 15 minutes rather than hours that we have on station. So essentially, we have to be envisioning the experiments need to be primarily autonomous. Also, they'll need to be compact. Sample return will be extremely limited. We'll be bringing astronauts back from the moon, but not much else. And so we'll rely a lot more on in situ analysis than being able to retrieve the specimens. And then we'd like to be able to command the experiments from the ground. And the higher radiation environment on Gateway relative to ISS means that the electronics will need to be radiation hardened. That drives us to still strongly consider the small sat payloads like BioSentinel as being readily adaptable to be used on Gateway. Looking beyond small sats, we need to look at adapting some of the hardware that's been developed for space station. A lot of great capabilities we've been able to take advantage of. They were designed with the ExpressRack payload interfaces. We'd like to adapt them to work on there's a set of new interfaces to be used in Gateway. So if NASA can invest in looking at that through the various programs we have, but that would be a great boon to the space biology community. Let's see. So one of the other things that we noticed as we looked across the different systems, there's a great deal of modularity built into these payload systems, and some of the modularity is across different platforms. So for instance, type 1, 2, 4, and 5 containers, which are essentially like cigarette pack in size, can be adapted and used in different types of centrifuges and incubators. And then also a lot of the hardware systems are have modularity built into them. So Tangle Labs and NanoRacks has U-size modules. BioServe has different inserts that go into their Sable incubator. TechShot also has developed a lot of different inserts, and the small sats take advantage of those 1U modules, different U-size modules. External payloads, I'll just say that yes, we definitely want to do experiments where we get to expose biology to the outside. It's not only of interest to space biology, but a lot of other disciplines like planetary protection and astrobiology. Looks like ESA is already developing some capabilities to study external payloads and have some autonomous servicing and analytical capabilities. So I want to take advantage of that. And then I'll just wrap up. As we look at these different platforms, it's important for space biology to think of a science strategy that leverages all these different systems and recognize each of these systems are going to afford some unique environments. If we can develop some reference experiments, perhaps do the same, use the same organisms, grow them in the same hardware system, but in these different radiation and gravity environments, it would be very valuable to look at and compare the data. We are seeing that certain things, the CubeSat type payloads can be adapted very easily to work across these different platforms. Looking more specifically at Gateway, we do want to take advantage of what's been developed for Station. So if there's ways we can update and adapt the ExpressRack payloads to work in Gateway, that would be tremendous. And on the lunar landers, we do want to take advantage of small sat systems, Biosentinel in particular. So I'll just finish up. So the punchline here is after we presented this information to Space Biology did take a further assessment of closer look at Biosentinel and essentially has decided that Biosentinel to work with Eclipse landers and is the LEA payload that's being developed right now is essentially Biosentinel adapted for that configuration. So I'll end right there. I'll take questions.
All right, thank you so much, Sid. We have about 1 minute to answer questions, so let's see how many we can get to. First question is, based on your assessment, do you think it would be possible to bring rodents on the lunar surface or maybe smaller animals?
Yeah, it looks like in terms of mass limitations, until we develop an environmental control life support capability, it'd be hard to bring rodents to the surface. That might be in the plans longer term, but initially I think we're going to look at smaller model organisms, and we'll be able to, but ultimately I'd love to see rodents that low Earth orbit.
Awesome. And one more question here. Are there any developments for robots or laboratory automation in space life science experiments in order to save crew time?
Yeah, so there are different innovations that are being looked at the payload level. There is not a dedicated program to look at this, though, and I think that would be something advantageous. Maybe something we can partner with STMD or AES. Those are technology development programs within NASA that could be really beneficial. But I think that would be, it definitely deserves more attention.
Wonderful. Thank you so much, Sid. We appreciate your time. That was a great talk. And will be Candace. She is a professor at the University of North Florida in the Department of Biology. And from 2014 to 2020, Candace conducted research as a senior staff scientist at the Space Biosciences Division of NASA, Ames Research Center, focusing on the mechanisms of spaceflight-induced bone loss. Her research at UNF aims to define the molecular mechanisms of cellular and tissue degradation resulting from spaceflight with a focus on the skeletal and cardiovascular systems. Take it away, Candace.
Thank you very much, Lavorka. Can you actually hear me and see my slides?
Instead of your
We
slides.
just lost your slides.
All right.
Okay, so we see
How
your
about that?
Perfect.
All right. Well, first and foremost, I'd like to thank the organizers for the opportunity to talk about some of the collaborative research that has been conducted among the members of the animal AWG. So before I get started on our interesting findings, I'd like to acknowledge the members of our animal AWG who have contributed to the work that I will be presenting today. And of course, NASA GeneLab team for processing the data sets that were used in this work. We already know from numerous studies spanning decades that spaceflight is indeed a profound stressor to organisms, tissues, and cells. And the reason for that, as we know, is that the space environment is composed of a unique set of stressors, which include microgravity and ionizing radiation, among other stressors. So the other stressors that we know are included in the spaceflight experience involves operating in an isolated environment, and as well as experiencing relatively elevated CO2 levels within the spacecraft on top of managing a demanding workload. So microgravity in particular is a profound stressor to the human body. And we know that exposure to microgravity, as shown here in this image, leads to a redistribution of fluid towards the head, which we call the cephalad fluid shift. Now, this cephalad fluid shift leads to an increased pressure in the head and thoracic regions. Now, another consequence of microgravity exposure is musculoskeletal unloading. Particularly the weight-bearing bones, such as that of the leg bones and the pelvis, experience a substantial loss in mass and mineral content. In addition, the postural muscles of the legs, such as the soleus muscle, are particularly sensitive to microgravity and indeed undergo atrophy, which is characterized by a decrease in mass and muscle cross-sectional area. So there are actually a number of ways by which spaceflight-induced muscle atrophy can be studied. So one of these, we can study muscles and muscle atrophy in space is by, of course, studying astronauts who have been deployed to the International Space Station or in earlier spaceflight endeavors in shuttle missions. However, since measurements on muscle function are relatively limited and technically challenging to conduct in space, one can also perform a bed rest study on Earth, which recapitulates some of the features, some of the consequences of microgravity on the human body, including the cephalad fluid shift and musculoskeletal unloading, which are experienced by astronauts. However, obviously, research in humans can have sampling limitations and ethical considerations. And that is why many researchers in the space biological sciences make use of rodent models. So that is obviously due to the relative ease of sampling tissues versus human subjects, and of course, the many similarities of rodent physiology with that of human physiology. So here, this image, you see the astronaut Scott Kelly handling the NASA rodent research habitat in the ISS. Now, since rodent spaceflight opportunities are relatively limited and resource intensive, one can also simulate the effects of microgravity using a number of Earth-based models, one of which is pictured here, called the Rodent Heimlimp Unloading Model, or HU model. So the HU model actually involves suspending the animal by the tail to induce musculoskeletal disuse of the hind limbs and a cephalad fluid shift, as you can see here. So today I'd like to share with you some of the findings from our animal AWG's recent publication. So in this study, our team aimed to identify candidate genes and pathways that play a role in spaceflight-induced muscle atrophy using a bioinformatics-based approach. Now, since I've already mentioned that mammalian spaceflight experiments and therefore omics data sets are fairly limited or relatively limited, we also aim to determine whether omics data from spaceflown invertebrate models can prove to be informative in gaining insight into the mechanisms of mammalian muscle atrophy during spaceflight. In order to identify potential genes and pathways involved in mediating spaceflight-induced muscle atrophy, we selected a number of transcriptomic data sets from spaceflight missions and microgravity simulations such as bed rest and rodent Heimlimp unloading. All of these data sets, by the way, are available from NASA GeneLab. Today, for the interest of time, I will focus on the results that we obtained from comparing RNA-seq data from two types of muscles from the 37-day Rodent Research 1 mission. Specifically here, we compared the soleus, which is a muscle that undergoes substantial atrophy during spaceflight, and the extensor digitorum longus, or EDL, which is relatively resistant to atrophy. So here I'm highlighting one of the interesting findings from comparing these two muscle types. What we found is that there was differential regulation of phosphodiesterase genes in these two muscles. Now, why is it an interesting finding? Well, we know that phosphodiesterases, or PDEs, are important regulators of cyclic AMP signaling, wherein these PDEs convert cyclic AMP to AMP. And cyclic AMP signaling is part of the secondary messenger signaling that is actually involved in a number of key biological processes, including the maintenance of skeletal muscle mass. In fact, in an earlier report, one group has shown that inhibiting one of the phosphodiesterases in muscle, specifically PDE4, prevents muscle atrophy. Now, interestingly, our team found that in response to spaceflight, mouse EDL, which is resistant to atrophy, showed downregulation of PDE2A. Now, in contrast, the soleus, which is sensitive to atrophy during spaceflight, showed upregulation of a number of PDEs, including PDE4. So these findings altogether suggest that PDEs may be a potential underlying mechanism for the differential sensitivities of the EDL and the soleus to unloading. Now, this also generates, the beauty of this approach is that this also generates a hypothesis that could be tested in future spaceflight missions. And specifically, the hypothesis is that will targeting these PDEs prevent muscle atrophy in space? Now, moving on to our second aim, we attempted to compare the transcriptomic data sets from space-flown fruit fly and nematodes, specifically C. elegans, with the same EDL and soleus RNA-seq data sets from the RR1 mission. And here I am highlighting some of the interesting findings from our analysis. Firstly, we found that the transcriptomic signature of C. elegans that has been exposed to spaceflight was actually more akin to that of mouse EDL from our R1 mission. Further, we found that the worm ortholog of the PDE4 gene was actually downregulated in C. elegans, just like what we found in mouse EDL from the RR1 mission. So this raises the hypothesis that perhaps PDE regulation could represent one of the shared mechanisms by which mammals and nematode models can resist muscle atrophy due to spaceflight. So interestingly, when we compared the fruit fly transcriptomic data sets, we found that the transcriptomic signatures of the larval stage of the fruit fly was actually more akin to that of the Mausoleus. And in particular, what's interesting here is that the shared differentially regulated biological processes between these two systems appeared to be consistent with a transcriptomic signature that promoted muscle atrophy. So in contrast, if you look at the transcriptomic signature of the adult fruit fly, that was actually more similar to that of the mouse EDL. And again, we found, which is really of interest to us, that the shared differentially regulated processes in these two systems or these two models share that of a transcriptomic signature that was more pro-proliferation. So in summary, our group found that there is a potential role of the PDEs or phosphodiesterases in mediating the response of muscle to spaceflight, and therefore could potentially be involved in mechanisms regulating muscle atrophy. We also found that the transcriptomic signature of spaceflown C. elegans was more akin to that of the mouse EDL. And interestingly, the transcriptomic signatures of D. melanogaster at the larval stage was more similar to that of the Soleus. And in contrast, the adult D. melanogaster transcriptomic signature was more akin to that of the EDL. So I'd like to end this talk by giving something really important to consider, that mammalian physiology has undeniably has undeniable differences from invertebrate physiology. However, using our omics approach here, our findings suggest that invertebrate models can actually be useful in anticipating some of the underlying molecular mechanisms for spaceflight muscle atrophy. So I'd like to end by saying that mammalian and invertebrate models are indeed complementary tools in understanding the responses of human muscles to spaceflight. Thank you for your attention.
Awesome. Thank you so much, Candice. We do have some questions for you here, but we have about a minute, so maybe we can answer one, maybe two questions. Have there been any studies of spaceflight effects on muscle satellite stem cells that are responsible for myocyte regeneration?
Well, there are just very few studies that have really looked at these in depth, but there is some evidence that at least in ground models, they do see a downregulation of the muscle stem cells or stem cell activity in these ground-based models. One of the challenges, by the way, of understanding the responses of muscles to spaceflight is that if we do live animal return, some of these changes some of the changes caused by spaceflight can actually be lost as the animals adapt to the 1G environment. And therefore, for future missions, it's really important to improve our ability to sample or to collect tissues or analyze tissues in flight. Perhaps technological advances would facilitate these endeavors.
Awesome. Thank you so much, Candice. So unfortunately, we are at time here. So we're going to thank you again, Candice, for your time and your awesome presentation. And we're going to move on. We will be moving on to the next speaker, who will be David Danko.
Thank you very much. All right, let me just get this presentation done. And all right, yeah, thank you. So my name is David Danko. I'm currently the Director of Software Engineering and Bioinformatics at Biosha, which is a microbial diagnostics startup. But before that, I was a PhD student in Chris Mason's lab, which is where I did most of the work that I'm going to be talking about today. And what I'm going to be talking about today, what I'm going to be presenting to you right now, is the twin study, which no doubt many people here are familiar with. I bet a lot of you have worked on this, know a lot about it. But I'm going to be talking about it in a of a different context, which is the context of the microbial built environment. I'm going to be talking about how the twin study is sort of a vehicle for us to study the microbial built environment and the good things we can do there. So the background a little bit to this work is that a lot of the stuff that I've studied, throughout my PhD, has to do with understanding biological interactions between humans and their built environment. So both how humans affect the biology of the spaces around them and how that biology then in turn comes back and affects humans. And one of the sort of the underlying paradigms I see in this, one of the ways I think about this, is that when you get to a sufficiently complicated ecology, so something like a building or a city, there starts to be very little distinction between a microbiome and just a regular biome. If you look at something just like size scale, the sort of literal definition of a microbiome, if you will, there's essentially no bright line where you can say that stuff smaller than this is a microbiome and stuff larger than this is a microbiome. Instead, what we have is this continuum, this ecological continuum. And because this is continuous, because there's no sharp breakpoint in here, it allows for the potential that there's small stuff that's having a dramatic impact on big stuff that's having a dramatic impact on small stuff, lots of interaction potential in a sufficiently complicated environment, which is where this idea of humans impacting their environment and being impacted their environment sort of derives from. Now, the NASA twin study, again, people here are probably largely familiar with the NASA twin study, so I'm only going to go through this very quickly. Two astronauts, Scott Kelly and Mark Kelly, agreed to go on a roughly 3-year study. During that time, in the middle of the study, Scott Kelly went to the International Space Station for a year. During that 3-year period, both of them were also extensively sampled. There were lots and lots of different samples taken from these guys, but the ones I'm most interested in here are oral metagenomes, gut metagenomes, and CD4 positive T cells. And simultaneously, also before Scott Kelly's flight, during Scott Kelly's flight, and after Scott Kelly's flight, there were environmental metagenomes taken from the International Space Station, work that I believe was largely led by Dr. Venkat's group at NASA. And this is sort of the underlying the basis, the data that I'm about to present here. And the International Space Station represents a pretty interesting locale to do a built environment study. And that's not just because, of course, the International Space Station is an interesting and kind of unusual environment, though of course it is. But it's also because it is a dramatically controlled environment. Unlike a terrestrial building, we can be very certain that Scott Kelly never left the International Space Station at any point during his tenure there. And we also know a lot about it. It's a heavily profiled system. There's a lot of data from it. For example, we probably have more information about the built environment of the International Space Station than we do about just about any building on Earth in aggregate. So it's a well-characterized system. It's a closed system. And that's what makes it so beneficial for the built environment study. The fact that it's in space is just like an extra cool factor bonus that makes it all that much more interesting. And so with that in mind, I want to use it to answer some sort of fundamental questions, if you will, about what human and built environment interaction looks like. And the first big question, maybe a little bit of an obvious question, is does microbial exchange occur between humans and their environment? And I want to distinguish two scenarios here that we're going to be able to tell apart with our data and hypothesis testing on. The first scenario, which you see represented on the slide here, is a scenario where no exchange occurs, where a human's gut microbiome is essentially static, it's not changing, there's no exchange with any kind of outside ecology or environment. And in that scenario, when you take repeated samples of this microbiome, you will at first find some new taxa, and that's because of stochastic loss in sampling. There's going to be stuff that you miss on the first sample that you find on the second sample, third sample, et cetera. But eventually, if you take enough samples, you're going to reach saturation. You're going to stop finding new taxa. You're going to have found everything that occurs in that microbiome. And except for the very, very slow force of evolution, you're not going to find new species if there's no exchange occurring with the external environment. The second scenario is one where there is exchange with some kind of broader environment, broader ecology. At this point, I'm not going to say that broader ecology is the International Space Station, but you'll just have to imagine it's some sort of amorphous external environment that is not the gut microbiome. And in that scenario, when you take repeated samples, you will still have that same drop-off at first as you sort of stochastically sample stuff that you missed the first time. But if you take enough samples, you'll reach a level where you're finding roughly a constant number of new taxa per sample. And those are samples that might have been exchanged with the outside environment. And critically, we can distinguish these two scenarios, the first scenario and the second scenario, with data. We don't necessarily need actually to know anything about the environment to distinguish these two scenarios. We can just look at them with time series themselves, see what these time series say. The difficulty, however, with comparing two different scenarios, understanding exchange occurs, is that we don't have an infinite number of samples. I can't just say, well, if we look at the 10 millionth sample, we're seeing this many new taxa. We, of course, deal with finite number of samples. And in fact, we deal with, even with these very good time series from the twin study, a very finite number of samples. So to get an effective comparison, we need a control case. And in this case, what we use for a control are roughly 250 metagenomic samples taken from exactly the same technical replicate, the same fecal replicate. So we know, because these samples came from the same underlying sample, that there was no exchange within the sample. It's the same sample. We sequenced it 250 times, assayed it a few different ways, but at the end of the day, it was the same exact sample, so there could not have been exchange with the environment. So we had these roughly 250 samples, we created some artificial time series, and we compared the number of new species we found in those artificial time series to the actual number of new species found in the real time series from Scott Kelly and Mark Kelly. By the way, Scott Kelly here is in blue with the code TW, and Mark Kelly is here in red with the code HR. What we find here is that the first few samples, the first few new samples taken in the time series, basically look the same between our artificial time series and our real time series. We're finding, statistically similar numbers of new taxa. But as we get out towards the later ends of new taxa, we find that it is drastically unlikely that the number of new taxa we find in Scott Kelly's and Mark Kelly's real-time series could be explained by random stochastic sampling of the same underlying sample. Instead, we see that this is likely evidence that there is new species, new genetic material being transferred into their gut microbiomes from some external environment. Now, if I'm arguing that there are new taxa being transferred into their gut microbiomes from an external environment, I ought to be able to show you at least one example of that, and this is what I'm about to do. So this is a graph of SNPs in Serratia protein maculans, and what you're looking at here is a graph of the variance relative to the Serratia protein maculans reference genome in Scott Kelly before, during flight, and after flight in both his feces and saliva. And there are a few things I want you to notice here. First, that there are two distinct strains here. You see one strain in his saliva, one set of SNPs in his saliva, and a second set of SNPs that's in his feces. And there also is a time series difference for this. The saliva strain is found before flight, during flight, after flight, basically the same SNPs. The fecal strain is only found during flight and after flight. And as you might guess, we do find that this fecal strain in one other place before flight, and that's in the International Space Station itself. We find all of the same SNPs that appear in Scott Kelly's feces during flight in the International Space Station. And this strongly suggests that there could be transfer from the environment to Scott Kelly. It doesn't definitively prove it, because of course there could be transfer from from some third source to both Scott Kelly and the space station, but it suggests a very strong possibility that there could be direct transfer from the environment into a gut microbiome of a person. And we find some other species that sort of match the same pattern, and the rate varies by different species, and interesting stuff like that as well. But that's all sort of details for the paper. Now, the next question, which becomes a little bit more, I think, sophisticated and more interesting here, is the question of whether this transfer from the environment provokes any kind of immunological reaction on the part of the person that is the recipient of this transfer. The scenarios we're trying to distinguish is whether this is totally like a subclinical, unnoticeable effect. These are just passengers that pass through someone's body, versus these are active microorganisms that provoke some kind of response in the body. And the way we have looked at that in this research is by looking at CD4 positive T cells, which, as many of you are likely aware, have a particular receptor structure that matches antigens presented by microbes. So microbes shed antigens, basically just protein fragments. And then T cell receptors will match those protein fragments and induce specificity for them. And what is important here, what is sort of critical to the technique I'm going to talk about, is that we know something about how the T cell receptor is structured. It's actually a very well characterized protein. And we can, with a high degree of statistical noise, admittedly, but we can estimate the protein sequences that a T cell receptor is likely specific to. This general technique of identifying motifs that a T cell can match to are called T cell exposed motifs. It was a concept originally introduced by Bream and Homan in 2015, with some good evidence there that this is possible and this exists. And we also have some additional evidence that this works in the wild, because when we took a very similar technique and applied it to COVID patients and control patients, we find many more T-cell exposed motifs that match the COVID genome in people that had COVID compared to our control samples of people that did not, suggesting that this technique may at least have some degree of usefulness for identifying targets of the immune system. And so we can apply this then to the broader case of an entire microbiome for the NASA twin study for Scott Kelly. And we can look at the T-cell exposed motifs that were found in Scott Kelly before, during, and after flight, and we can map them to microbial taxa. And because these are so short, because these are just like 5 amino acid sequences, the T-cell exposed motifs, it's relatively hard to say exactly what taxa each of them map to. So we just treat these as sort of sets, and we try to find how much overlap there is between sets. And what we find is that when we compare the actual T-cell exposed motifs that we find in Scott Kelly, we take random subsets equal to the number of species in the International Space Station, we can create a background distribution. And the actual species found in the ISS are in general more similar to the T-cell exposed motifs that we find in Scott Kelly before flight and after flight, but they are well within what you might expect by just random variation in the distribution. However, during flight, we see this massive upshot where there's much, much higher similarity between Scott Kelly's T-cell exposed motifs and the microbes in the International Space Station than would be expected by random chance, or that are seen before flight or after flight, which we believe is preliminary evidence suggesting an immune response that is specific to the environmental microbes in space. We can also see that over time with this sort of gradually increasing in the space station, but it, in general, being much, much higher than Mark Kelly, the purple dots here, time series was at any point. And with that, I'd like to wrap up. This work is the product of a lot of different people here, but the ones that I particularly want to thank here are Danielle Besson, who's also one of the organizers of this conference, Chris Mason, my PI, and Dr. Venkat, who was sort of the NASA liaison I worked with for a lot of this data and was certainly very supportive and helpful, as well as many, many other people that made this all possible. So with that, I can take some questions now, and thank you for listening.
All right, thank you so much, David. We have some questions here. Why is the immune response only specific during flight, since the variant is still present after flight?
So the immune response I was showing wasn't specific to Serratia proteomaculans. It's just to the taxa we find in the ISS generally. So it actually, that's an interesting question. I haven't, to be honest, thought to look at what happens to the immune response to Serratia proteomaculans, particularly before, during, and after flight. But yeah, you would expect, I think, to still see something. That said, if Serratia proteomaculans is tolerated and is retained after flight, it's possible that there's also not a strong immune reaction to it at all, because otherwise you would expect the immune system might be clearing that species.
Awesome. Another question is, did you compare metagenomes from the Astronaut and the ISS and compare the genetic diversity of the samples?
Yes, we definitely did, but it's not super easy to compare that kind of thing because different metagenomic techniques in processing can result in such different diversity metrics. There's a lot of systemic problems to deal with metagenomics that it's hard for me to give a real answer. You tend to get more lower abundance stuff in environmental microbiomes. The space station was different, but was no real exception to that. But they were sampled with such different techniques. I don't want to say definitively that that's true and that all the microbes we find are necessarily viable.
Awesome, David, thank you so much for your time. We are at time, so I apologize that we cannot get to the other questions, but we do have another speaker. We have our next speaker here before we break, and the speaker is Aranza Manzano. And she obtained her PhD in biology from Spain under the direction of Dr. Javier Medina and Raul Herranz. The main line of her research in space plant biology is primarily focused on the effects of gravity levels on the development and growth of Arabidopsis thalia seedlings, both at the cellular and molecular levels. Aranza, go ahead and take it away.
Okay. Hello, thank you. Can you see my presentation?
Yes, looks good.
Okay, thank you. Next, I am going to present some results from Javier Medina and Raúl Herranz's laboratory obtained for the space experiment, in particular, how some stress-relative mutant plants may show an improved molecular response to spike flies and Mars gravity level. In root experiment carried out at ESA during the Cervantes mission and in experiments performed in ground facilities, the effect of microgravity on the meristematic cells of 4-day-old Arabidopsis thaliana seedlings growth in darkness was studied. The results showed that these experimental conditions produce seedling growth stress. At cellular level, increased cell proliferation but decreased cell growth, and therefore cause an imbalance in the two most important processes that support plants' growth and development. Nucleolin is the most abundant multifunctional protein in the nucleolus, involved in different steps of ribosome biogenesis, and also has an important role in other processes such as DNA replication, recombination and repair, DNA, RNA metabolism, and cell proliferation. In Arabidopsis thaliana, two genes encoding nucleolin have been described. The NU1 gene is highly and ubiquitously expressed in all tissues under normal growth conditions, and the NU2 gene, whose expression is restricted to specific tissues and organs during plant development. Our interest in these genes is based on the fact that its expression increases under stress conditions, so we hypothesize could be involved in the response or adaptation to these conditions. Disruption of the NU1 gene induced expression of the NU2 gene lead to severe effect on plant growth and development. In contrast, the disruption of the NU2 gene causes a nearly normal phenotype with a delay in flowering. Due to the important role that nucleolin has in cell growth and that this process was seriously altered in previous laboratory experiments, the two mutant nucleolin lines were included in the seedling growth experiment with the goal of knowing the role of this protein in adaptation to a spaceflight environment. In this experiment, the plants grew for 6 days inside the ENCAs at two constant gravity levels, microgravity and Mars gravity. The first 4 days they did under a photoperiod lighting regime, and the last 2 days, one part of them was kept in darkness and the other part was photostimulated with red light. The dark condition was applied to know the effect of the lack of gravity, as the plants didn't have any signal that direct their growth, and photostimulation with red light was applied to study to what extent this signal can compensate the deleterious effect of microgravity. Since in addition, is known that in particular the red light stimulates ribosome biogenesis and cell proliferation and increases the expression of the nucleolin gene. After the growth period, the seedlings were frozen to perform transcriptomic analysis of their fees to carry out a microscopic analysis to determine nuclear function. The photos captured from the ENC cassette after the experiment show neither of the two gravity levels or each of the illumination produced serious alterations in the growth of the seedlings, since their growth is equivalent to the plants on the ground control. There are also no difference between the mutant plants and the wild type. On the other hand, the principal component analysis show a clear cluster of the sample based on light and gravity level. There is a clear difference between dark samples and red light photostimulation samples, and respect to gravity level, there are two clear groups. The microgravity samples differ from the samples subjected to the Mars gravity and ground control. We are clustered together. Looking at the global RNA seed results in microgravity and darkness, the wild type includes the expression of the both nucleolin genes, especially NU2, which is in agreement with the fact that is genotype with a greater number of the deregulated genes. Functional analysis show that up-regulated genes are mainly involved in starvation, seed termination, and lateral root formation, and that down-regulated genes are related to ion transport, a different metabolic process. These results are compatible with a survival response to an stress environment, such as this one, as a consequence of the lack of any tropistic signal combined with the severe decrease in photosynthesis and gas exchange described in microgravity. On the contrary, in the NUT2 mutant, there is no increase in the expression of the nucleolin 1 gene. This could be related to the fact that this line is the one with the lowest number of the genes differentially expressed in microgravity and darkness, and that, in addition, are not involved in essential processes for plant survival. The up-regulating genes are related to the third world and different metabolic processes, and the down-regulating genes participate in the immune response and respond to temperature. Therefore, in contrast with the wild-type response in the NUT2 mutant, lacking the NUT variant involved in processes or respond to stress and defense can be an adaptive advantage to the most challenging experimental conditions. Finally, the condition of microgravity and darkness produce a reduction in the expression of the NUT2 gene in the NUT1 mutant, which is linked to the fact that in this genotype most of the differentially expressed genes are down. Furthermore, these genes are involved in reproduction and growth processes, which indicate that in this mutant, this experimental condition enhances the negative effect of NUT1 gene dilution, providing less acclimatization capability than wild type. This conclusion is also supported by analysis at the cellular level, where both proliferation and cell growth have the lowest value. When the plants are photostimulated with red light in microgravity, the number of differentially expressed genes shared by the wild type and the NUT2 mutant is very high. This may be due to the fact that under this condition, both genotypes behave the same as the expression of the NUT1 gene increase in both. Also in wild type, the expression of the NUT2 gene increase also less than in the previous conditions. Among the genes common to both lines, some genes with a regulating expression are related to transport, cold acclimatization, and down-regulated genes are involved in photosynthesis, immune system, and ribosome biogenesis. So red light has a positive effect on plant ability to grow under microgravity because above some categories that imply a clear survival response, but some processes are still altered so that it is not be able to fully compensate for the effect caused by the lack of the gravity. Further, the similarity in the response of wild type and NUT2 mutant in the presence of red light suggests that the express program related with the nuclear protein NUT2 expression is not activated in microgravity under positive photostimulation. Unlike what occurs in darkness, in microgravity and red light condition, the NUT1 mutant shows an increase in the expression of the NUT2 gene together with the lower number of the gene with differential expressions, which are not involved in essential processes for its growth. That is, red light stimulate growth and proliferations. Results that are also corroborated by microscopy analysis parameters. After microgravity effects are disclosed, we approach mass gravity effect in darkness. Change in nucleolin gene expression is not observed in wild type and in the NUT2 mutant. There is only a slight increase in the expression of NUT2 genes in the NUT1 mutant. In addition, the three genotypes share a large number of the differentially expressed genes. Most of the upregulated genes are involved in the response to different stimuli and in the regulation of the different biological processes, and the down-regulated genes have a function in transport and photosynthesis. The results indicate that the three lines are able to detect and promote an adequate adaptive response at partial gravity level. But despite this, important processes and plant structure are affected. Photostimulation with red lights in Mars gravity does not change the expression level of the new 1G in either the wild type or the new 2 mutant, but as in microgravity and darkness, there is an increase in the expression of the new 2 gene in wild type. This is accompanied by the fact that in the new 2 mutant is the genotype with the lowest number of differentially expressed genes and the wild type with the highest number. Most of the differentially expressing genes in the wild type are regulated and are involved in reserve of mutant and respond to stress. The result could indicate that a weak gravitopic signal together with a red light photostimulation produce an exaggerated response in wild type plants due to the conflict of the two signals. So the use of new 2 mutant may be beneficial in this part gene level environmental. Finally, we want to express that this condition an increase in the expression of the new 2 gene in the new 2Y mutant. This mutant have a regulating mainly involved in heat response and down-regulating gene involved in different metabolic process. All of it is in agreement with the possible involvement of the new 2 protein in the heat stress and with the effect related to the lack of the new 2 protein. The large increase in new 2 gene expression in this mutant in this condition is also reflected in the cell growth parameters. While cell proliferation rate is similar, the larger nucleoids are corresponding to the mass gravity and red light. To summary, the main conclusion of the studies are mutant with reduced stress response could be good candidate for use in the future life support system under the more stressful spiral condition. New to mutant growth and development is equal to that the wild type, but without altering the expression of gene involving in process essential for plant survival. Mass gravity level is enough to trigger an appropriate adaptive response by increasing the expression of the gene involved in biological regularity processes, and photostimulation with red light had a positive effect on plant growth in the experimental conditions studied, especially in the new 1 mutant, in which it promotes cell proliferation and cell growth. Now, Raúl will tackle about GeneLab. Sorry.
Well, hello. Thank you, Arantxa, for the presentation. If you can go to the next slide. I just want to I don't know if you can do it. Yes, well, I just want to emphasize that some of us, including Arantxa, but also Stefania, the first speaker of today after Sylvain, and also the following one, Joseph, are European members, also participating in the GeneLab Analysis Working Group. And for those of you that are working in Europe and want to participate in a different working group, we have the ISA topical team, if you can go to the next slide, in which we coordinate our activities at the European level. I don't know, Arantxa, if you can go to the next slide. So there are several peoples also. We are also coordinated with the ISSOP consortium because we are also members of this international activity. And well, the important thing is that you visit our webpage if you want more information about us, and also if you want to know what kind of expertise we have and in which way we can collaborate further. And if you can go to the next one. Well, this is just for you to check that you meet the requirements if you want to join the Spatialomics topical team. In fact, to be interested in the Spatialomics research field and to work in a European laboratory. And that's all. Thank you, Arantxa, for these seconds of your time. Thank you very much.
Wonderful. Thank you, Arantxa and Raúl. So we do have some questions here. Maybe we can use up 2 minutes of our break to answer questions. The first one is, can you please summarize the significance of your results for growth of plants in the Mars gravity environment?
Yes, okay. For the free genotypes, the more important results in the Mars gravity is in In the darkness conditions, the response of the plant is correct to adaptive response correct, and the severe effect observed in microgravity and darkness don't exist in Mars gravity.
Awesome, thank you. The next question is, can you see differences between tissue types in growth? Example, are roots affected differently than the leaves?
No, in this study, the RNAs was straight for whole plants, so for whole seedlings. Don't separate roots and shoots.
All right, and our last question here. The root media in space cannot provide oxygen to the plant equivalently to that on Earth. If the root module runs low on oxygen in space and root-shoot communication is key, how do you control for spaceflight hypoxia in this system?
Okay, this is true. The plants in seedling growth grieve in the European modular cultivation systems. These are hardware controlled and provide the plants for temperature, humidity, and atmosphere. So controls atmosphere is the manner of avoid hypoxia. Is the one.
Wonderful. Thank you so much, Aranza and Raul. We appreciate your participation, and we are going to take a break until 1:10 p.m. Pacific time. So we will see you all shortly for our final presenter, Joseph Borg. All right, welcome back, everybody. We have our final speaker of the day today before we break out into our AWG meet and greet breakout sessions. And today's speaker, we have Joseph Borg. He's a professor at the University of Malta. And Joseph initially studied applied biomedical science at the Faculty of Health Sciences at the University of Malta, and subsequently followed a specialized training in the molecular genetics in molecular genetics and hematology. Joseph's current research interests with the Department of Applied Biomedical Science continue to focus on the molecular genetics of developmental globin gene switching and control. Together with his Borg group, he conducts extensive research on KLF1 erythroid specific transcription factor, utilizing both genomic and transcriptomic approaches to in vivo and in vitro models. Take it away, Joseph.
Okay. So can you hear me like this? Am I being heard?
We can hear you.
Okay, because for a moment I thought I had some problems with the sound. So thank you for the introduction. I just came back from another activity or event also talking about Project Mallet. So this is my second one this evening, and I will try to make it as lively as possible as well. So yes, so my talk today won't be too much about the data, too much about results, but more about the very first mission that departed from Malta all the way back to Cape Canaveral and its first mission on board the International Space Station. So yes, so actually it has been a year now, because the idea, the concept was developed around about October, November 2020, and in just under a year, or just about a year, this whole idea was built up to a point where the whole experiment was set up, prepared, it was designed, and together with our colleagues based in Belgium, Brussels, which are called Space Application Services, basically there was a whole checklist of events or activities that facilitated us to prepare this project from idea to launch. So what is Project Mallet all about? Okay, so essentially there are two important aspects to Project Mallet. Okay, first of all, the real science under Project Mallet is all connected with diabetes, especially diabetes mellitus type 2, and very specifically those patients who develop wound ulcers at the extremities or at the bottom of the feet. As some of you might know, these wound ulcers, there is a tendency that become infected, and these infected wound ulcers, conventionally, a wound swab is taken. This is taken to the lab, to the bacteriology laboratory, and from there they are tested for what type of bacteria they are, what they are sensitive to, and what they are resistant to. So my PhD student, Christine Gatt, was also a biomedical scientist. She works at the bacteriology laboratory. She is actually reading for her PhD at the University of Malta. And very specifically, she's studying the genetics of bacteria that infect wound ulcers. So she has hundreds of patients that she is studying. But for Project Mallet, we had the opportunity to study 6 patients from a human skin sample, complete with the microbiome, and we send this on board the ISS. Of course, the idea was that besides the conventional testing and besides the normal testing done in a laboratory at hospital, we wanted to understand or identify how these microbiomes, how these microorganisms in the context of a human skin sample, how they would adapt, behave, evolve in the context of microgravity and a relatively high level of solar radiation, keeping in mind that this was a very first design project stemming out from Malta. Yeah, so biomedical science, as most of you, is a conventional or a classic approach to ask questions and produce results and answers. So the whole microbiology is done by using conventional tools and techniques. But of course, for Project Mallet, all this had to be condensed in a miniaturized version. Yeah, so as you can see there on the right-hand side, that is a look inside the cube itself, where there are 6 sample cuvettes. These were prepared with a specialized buffer, and each cuvette contained the human skin sample with microbiome. They were sealed and they were kept indefinite at ambient temperature. On the left-hand side, you can see a typical blood agar and all the microorganisms that grow after streaking a wound swab. Yeah, so first of all, the difference between conventional microbiology and using next-gen sequencing. So the idea is that we want to use DNA-based tools to uncover all bacteria, all the microorganisms that are present in a tissue, and bypassing, if need be, the culture step. And this idea, the conducting next-gen sequencing or genome-based techniques, is to be employed for the samples that are present here in our labs and for the same samples that have been sent to space and back. Yes, I told you that Project Mallet is about 2 things. So of course, we all love science, but we also love to engage the young to appreciate science, and possibly, maybe sometime in the future, they also choose science as their profession, career, or some sort of a hobby that can pay off large dividends. So this is one example where we went to schools, we showcased the Project Mallet, sending things to space, and we told them that if you can send a message from space down to Earth, what would you do? What would you write? And you get all sorts of ideas and pictures, and it was a very interesting sub-project under the Mallet experiment. Yeah, so go back to the cube. There were, of course, phases of assembly where the cube itself was being prepared. The Raspberry, it was powered by a Raspberry Pi module. It had 2 cameras that look at the cuvettes in place. The samples were taken from Malta to Belgium, Brussels, because that is the place where the final integration and preparation was performed. On the right-hand side, you can see there the 6 sample cuvettes being inserted into the BioCube. And finally, this is all set and ready to close. So this is a closer image of the human tissue samples. There you can see in each cuvette with the buffer itself, and just before closing the cube. And this is a tiny movie clip showing perhaps in greater detail or a closer image of the cube itself. Okay, and there at the top you can see also the SD card that contained messages and audio files and video from the kids in Malta. This is a typical software view of the software that we use to connect to the cube, to access the cube, to monitor the temperature. We could play with lighting. We could also measure some form of sensor data, and basically it was our gateway to the experiment on board the ISS. Yes, so in Belgium at Space Application Services, there is an exact replica of a platform called IceCubes for international commercial, for the commercialization platform. And this replica is found within space applications in Belgium. And the reason being, so all testing can be done on site, which is a faithful representation of the same platform type that is found in the Columbus module in the ISS. So this was the very last picture taken of the cube before preparation to depart from Brussels to Kennedy Space Center. So these three images is showing the packaging of the cube itself and on its way to America. Yes, and finally, after arrival on board the ISS, the main astronaut who handled the cube was Thomas Pesquet from France. I'm not sure if you can. Yes, let me see if you hear you.
I'll move to 4 for the group conference.
So this is a tiny clip.
And Thomas, we are very much grateful for your support today. We also took note of your juggling skills, so thanks for the entertainment. And the group conference is ready on Space to Ground 4.
All right, thank you. It was fun to do, and it's good to see those new players in space, Malta and others. So congratulations to them for their CubeSats, and moving forward.
We copy that, and have a good day.
Okay, so those were the first and last words of Thomas Pesquet on handling the BioCube itself during the assembly, and of course it served to make Malta's history for the first time, hoping there will be many more to follow. And this is the cube itself, physically located on board the IceCubes facility, ready to be pushed back in the drawer. And basically the samples were left untouched for the whole duration of the SpaceX CRS-23 mission, which was more or less 30 days. After the 30 days elapsed, so the experiment was, of course, brought down back to Earth, splashing down off the coast of Florida. The cube was sent back to Kennedy Space Center, from there packaged back and sent back directly to us here in Malta. We were also very happy and excited to see that parts of this project also made it to the highlights of the space station. So we were all very happy about that. And finally, the last slide or two. So what are we doing right now? So first and foremost, this is a biomedical science and the genomic study. So we will be doing comparative genomics. Right now, the DNA has been extracted from bacteria, because yes, from each 6 individual samples that we sent, bacteria have grown on agar plates. We know their species name, so there are different microorganisms that grew, actually a mix of different microorganisms that grew on the agar plates. But now that is not all. So we want to dig deeper into the genetics and the sequence of the microorganisms to see if there are any meaningful changes, and if there are also any significant changes between different types of microorganisms. Having the human skin DNA present as well, we are also embarking on a genome sequence, a full genome sequence of the patient's DNA that traveled to space. Right now, we're performing 16S typing. However, there is a plan and an idea to do a proper metagenomic sequencing of the isolated microbiome. And there is also a query whether we can conduct metabolic profiling on the available sample material. So we hope that in the coming couple of weeks, you will be hearing more about this in the public, in the media, but also in an official manner by means of publication. Some important and special thanks before I conclude this small presentation. The Ministry of Foreign and European Affairs were the primary driver of the project, where they believed since day one. Of course, it didn't take much to convince them how much this project is foreign and international, and this is because we're dealing with space. Two important and very close colleagues of mine that they've come to learn through being an active member of NASA GeneLab and also through Project Mallet is Afshin Beheshti from NASA Ames Research Center and also Chris Mason and his lab at Cornell, whereby the initial discussions were also dealing with blood science, and slowly we also progressed to this project, and they are helping in some aspects of the genome sequence and the analysis. Then important people in Malta, the vascular surgeon who obtained the samples and his team, the people at Arcafort, which is a technology company and focus on connectivity. So they helped us ensure proper connection between Malta and the ISS experiment. And Evolve is a biotech company here in Malta that assisted us monetarily as well by offering its monetary support to procure or purchase the important reagents and the biomedical supplies to progress with the experiment. Okay, so that was the last slide. I will stop the share.
Awesome. Thank you so much, Joseph. So we have a couple of questions for you here. And the first is congratulations on being Malta's first mission to the ISS. As the first PI to have an experiment for Malta, what would your advice be for students or PIs to propose space experiments?
Okay, so I would not lie. The initial drive was very much a personal drive or a personal ambition. However, keeping in mind that so initially, if you remain sort of encapsulated alone, it's very difficult to penetrate or to make it real. However, the some people that actually work in the sector, a couple of which I have just named, like Afshin and Chris Mason, and there are many others who form part of NASA GeneLab and also the ESA Space Topics topical team. I would say reach out, talk to them, discuss your idea, ask for feedback, and basically it can really be done. It's really, I mean, it takes perseverance, but it can be done.
Wonderful. And we have another question here. Somebody is wanting to ask if you can provide more information on if you will be looking at metabolomics and perhaps when.
Yes, indeed, metabolomics is in the pipeline, so to say. We have sort of meticulously split the sample into different aspects. The original and the first idea was definitely to look at the genome profiles and also a full genomic profile. However, samples have been preserved for metabolic profiling, and we intend to do that not in Malta because we are not heavily equipped to do this, but we have discussions either with a private sector company or else with colleagues also based in the States. If this will be done very fast in the span of the next 2 months. So we also hope that all genomic-based data and experiments will also be concluded either by the end of this year or at least in the first quarter of 2022.
And we have another question here. Due to the limited amount of samples and biomass, how are you optimizing the sample processing for the multi-omics assay proposed?
True. That is a very legitimate question. Indeed, the sample material, it is what it is. Luckily, and here we thank the sort of the vascular surgeons, right? The human skin sample that was provided to us was not small, was not little. And in fact, the sample at hand had to be split at least for 3 different time points. And these 3 time points are day 1 or day 0, so when the sample was received in the lab, and then the next 2 time points are the sample kept in Malta and the sample sent to space and back. We had a modest weight or sample weight in that sense, but basically we went by division, right? So we had divisible numbers that were common across the board, and we will try to capitalize as much as possible in obtaining. So that is why, if we are able to conduct a full metagenome profile, we will try to opt for that in an optimized manner, as opposed to having to do smaller cycles of selected gene segments or PCRs or so forth. So I am a strong believer that for multiomics, you try to conduct a full genome scan, maybe full transcriptome scan that also allows us. Metabolomics is being discussed, and we hope that all of this can serve as a good comparative genomic study between the different omic states. Of course, I am this is a very much learning curve in progress, and we hope that the lessons learned now will improve the next and future experiments that will send from Malta.
Wonderful. Thank you so much, Joseph. We are right on time here. So I thank you so much for your participation and your wonderful talk.
Thank you. Thank you.
Awesome. So that concludes our speakers for today. And right now, we will be breaking out into sessions for the breakout sessions